Good day, students. Welcome to JSS 1 Basic Science. This is the very first lesson and it lays the foundation for everything that follows. We are going to learn about science itself, what it is, how it works, and how scientists investigate the world through the scientific method. By the end of this lesson, you will be able to define science and state the branches of basic science, describe the scientific method step by step, distinguish between observation, hypothesis, experiment, and conclusion, identify science equipment and safety rules in the laboratory, and explain why science is important in everyday Nigerian life. Let us begin. What is science? Science is a systematic and organized body of knowledge about the natural world, obtained through observation and experiment. The word comes from the Latin scientia, meaning knowledge. Branches of science: biology, living things; chemistry, matter and substances; physics, energy and forces; earth science, geology, meteorology, space science, astronomy. Basic science at JSS combines all these branches. Importance of science. Science explains natural events, why rain falls, why food decays, how diseases spread. Science drives technology, electricity, medicine, phones, transport all come from applied science. Science helps farmers improve crop yields, doctors treat diseases, engineers build bridges. Nigeria's economy depends on science and technology in oil and gas, agriculture, and manufacturing. The scientific method: 6 steps. Step 1: observation. Notice and describe something using your 5 senses. I noticed that the pond near our school turns green every August. Step 2: problem/question. Ask a question about what you observed. Why does the pond turn green in August? Step 3: hypothesis. Make an educated guess, prediction, based on what you know. The pond turns green because tiny plants, algae, grow in warm, wet conditions. Step 4: experiment. Test the hypothesis, change one variable at a time, the independent variable, measure the result, dependent variable, keep everything else constant, controlled variables. Step 5: result/data collection. Record what happened using tables, graphs, or measurements. Step 6: conclusion. Decide if the hypothesis was supported or not, communicate the findings. Note: if the hypothesis is not supported, you revise it and experiment again. Science is self-correcting. Science laboratory equipment. Beaker, cylindrical glass container for holding and heating liquids. Bunsen burner, gas burner for heating. Safety flame equals blue, luminous flame equals yellow, less hot, more soot. Test tube and holder, small glass tube for chemical reactions, use holder when heating. Conical flask, Erlenmeyer flask, triangular flask for mixing liquid safely. Measuring cylinder, for measuring liquid volumes in milliliters, ml. Thermometer, measures temperature in degrees Celsius, degrees Celsius. Hand lens, magnifying glass enlarges small objects for observation. Microscope, magnifies very small objects, cells, microorganisms, many hundreds of times. Tripod stand and wire gauze, support a beaker or flask over a Bunsen burner. Weighing balance, measures mass in grams, g, or kilograms, kg. Science laboratory safety rules. Never eat, drink, or taste any substance in the lab. Always wear a lab coat and goggles when handling chemicals. Read all labels before using chemicals. Never smell a chemical directly, waft the smell towards your nose with your hand. Report all accidents, spills, cuts, burns, to the teacher immediately. Tie back long hair, remove loose clothing near flames. Never run, push, or play in the laboratory. Know the location of the fire extinguisher and first aid kit. Variables and fair testing. A fair test changes only one variable at a time. For example, testing whether sunlight affects plant growth, change light, independent, measure growth, dependent, keep water, soil, temperature the same, of controlled variables. Bias in observation, Scientists must record what actually happens, not what they expect to see. Results must be reproducible. Other scientists should get the same results if they repeat the experiment. 3 examination questions. Question 1. A student noticed that her plants near the window grew taller than those in the corner. She decided to investigate. Write a hypothesis for her investigation. Hypothesis: plants that receive more sunlight will grow taller than plants that receive less sunlight. Reasonable hypotheses vary. A hypothesis is a testable prediction. Question 2. Identify the variables in this experiment. A student tests whether adding fertilizer increases the yield of tomato plants by growing 10 plants with fertilizer and 10 without, or receiving the same amount of water, sunlight, and space. Independent variable: change presence or absence of fertilizer. Dependent variable: measured yield, number/mass of tomatoes produced. Controlled variables: kept constant water amount, sunlight, space, soil type, plant variety. Question 3. State 3 safety rules to follow when heating a liquid in a test tube. A. Point the open end of the test tube away from yourself and others. B. Hold the test tube with a test tube holder, never with your bare hand. C. Heat the liquid gently, shaking/moving the tube, and never seal the top. 3 common mistakes. Mistake 1. Confusing hypothesis with conclusion. A hypothesis is made before the experiment, it is your prediction. A conclusion is made after, it is what the results show. You cannot hypothesize after you have seen the results. Mistake 2. Changing more than one variable. If you want to test whether temperature affects reaction speed, you must keep everything else the same. Changing 2 things at once makes it impossible to know which one caused the result. Mistake 3. Writing "I proved my hypothesis" in conclusions. Science never absolutely proves anything. Results support or do not support a hypothesis. New evidence can always change scientific understanding. Summary. Science: systematic knowledge about the natural world obtained through observation and experiment. Scientific method: 6 steps: observation, question, hypothesis, educated prediction, experiment, change one variable, results, record data, conclusion, support/reject hypothesis. Lab equipment: beaker, test tube, Bunsen burner, microscope, thermometer, measuring cylinder. Safety: no eating/drinking, lab coat and goggles, report accidents, waft smells. Variables: independent, changed, dependent, measured, controlled, kept constant. Fair test equals 1 variable changed at a time. For homework: A. List 3 things you observe in your daily life that could form a science investigation. B. Design a simple fair test to find out whether salt affects how fast water boils. C. State 5 lab safety rules. Attempt a quiz on your LMS. Keep exploring the world around you. Visit Litech Academy at academy.litech.com.ng. Until next time.